So our scrappy little tiny but mighty community raised $180,000 to put into Andrew's lab. That research turned into $2.3 million worth of investment from the US government, but it's not US based, right? This is science. Science is global. So everything Andrew finds out, everything the Chew Lab finds out, it's going to be important for our whole community across the world. So Andrew doesn't like to brag about stuff, so we'll brag for him. So he is going to provide an update, and then we'll hear from Clement, and happy to take your questions.
I want to thank every family, every single one of you for coming. It's a beautiful city. We're actually facing the Michigan River. It's beautiful. I want to thank the organizers, in particular Susan, for putting this together. And I want also, and the organizers are many, so I just want to, Susan, you represent that organization. So I also want to thank the hosts, Dr. Parada, and well, he didn't do most of the work. Caroline did most of the work. So I want to thank you for that. I think I didn't reply Susan's email in time, and you made up the title for my talk called the Research Update. Yeah, so I'll go with it. All right. Recognizing we're in Chicago, so I'll just put up there. So sleep is important. It plays important roles in a number of processes, including energy rebuilding, information processing, memory consolidation. And in the past 10 years or so, there's this increased recognition that it's crucially important for flushing toxins in the brain. Okay, that happens only during sleep. Sleep affects virtually every single cell and system in the body. Okay, so sleep problems are common, unfortunately, in a modern society, in the general population, but more so in patients with different conditions. Yeah, I'm sure you're not tired of me saying sleep's important, but I want to make a more fundamental comment, note here, that is, sleep problems, are reflected at the single cell level. So there's something wrong fundamentally in the cells. That's why, so sleep problem is a reflection of cellular dysfunction. So we want to find that out. Maybe a disappointing kind of statement here. Despite decades of research in seizure, sleep, we still don't have a good understanding where it's regulated and what genes regulating these processes. Therefore, you can see the challenges of treating and especially curing these conditions. So we want to bear in mind that in order to achieve that goal, we need to understand at the fundamental level in cells, in tissues, and as we're a whole body. So at the what's called in systems level. Okay. So I just want to emphasize the mTOR function is multifaceted, and SKS is a multi-system disease. So every single cell has this protein, and every single cell needs this protein to detect the energy level in the blood, and to then tell what the cells need to do, either grow, proliferate, or require more and tell the brain to eat. Okay, so this protein is essential, and too much, too little. As we do research, too much, too little, either is not good. So you have to have a fine balance, and those balance are time of day dependent. So for us in humans, mTOR activity is higher during the day and lower at night, and that pattern is important. All right, so research update. So from the 2013 to 2018, we discovered that mTOR regulates the light input to our brain. So we're a day-night human being. The light input is regulated by mTOR. And at the cellular, tissue, and organismal level, mTOR also regulating circadian rhythms. The 2 a.m. versus 2 p.m. are definitely different. So this protein also regulates the circadian rhythms, including the sleep-wake behavior. In a very fortunate, unfortunate, circumstance, we formed collaborations with Cincinnati Children's, with other fantastic clinicians and researchers, as well as with the SKS Foundation. So I was proudly to say that I was the founding member of that committee foundation. So with support from the SKS Foundation, we developed cellular models to test. So this is basically the hardcore question. When you have a so-called mutation, a variant, does this variant have a consequence? So that's the fundamental question we want to close the gap between genotype and phenotype. Does this variant have any consequence affecting the protein activity, affecting much higher level functions, such as metabolism, such as sleep? And as you mentioned, I want to thank the SKS Foundation. We use this funding to develop these assays, and we were given the grant. So my collaborators here are John Hoganesh and Dr. Prada right here. So thank you I want to really thank the people who actually did all the work. And so a bit kind of update here. So from 23 to 2024, we developed preclinical cell models. We have pretty much for cell models for all the variants, and we were able to test the consequence of that so-called mutation or variant on the proteins. Does it affect the activity of the protein, and does it affect the activity of the cell? Okay. And we developed 3 mouse models based on their activity levels from high to low, and also based on the prevalence. Like the middle one, E1799K variant, is present in virtually 50% of all patients in the SKS. So we want to model this in mouse models. And I want to just shout out for the fly people. Okay, you can generate fly within months. We generate this mouse within years. Also an update that we have a manuscript in place under review, and I'll go share a little bit about the current research. So this paper is under review, and we had a pretty difficult time publishing it in high impact journals, meaning that those journals that are top-notch journals for different reasons. This is not clinical. This is not fundamental genetics. So it's a combination of both. Those journals have a difficult time deciding what to do with it. But I want to emphasize what we did here. What we did is to develop the assays to actually test the consequences of a variant in the biochemical protein level as well as in cells. And we were able to use sleep-wake as a biomarker, meaning, in Jack's case, right? Is he sleeping well? If he's sleeping well, then the dosage must be good, okay? So this is the first time, and we can actually use this very good drug to kind of improve sleep. Now, so far, there's not a single drug in the medicine that can actually target sleep. Okay, so basically, the drugs are sedatives, hypnotics to actually put you down to sleep. So that's not a drug to cure sleep. That's just make it knock you down, to sleep. You drink alcohol, like 1 bottle of vodka, it's the same effect. So it's not targeting sleep, to say. So I want to thank all the people on the authorship. It's a huge collaboration among all the clinicians and researchers. Several people are here. Caroline, Lindsay is here. Darcy, and colors. Very quickly, so we were able to model every single variant in the cell. We can monitor whether that variant actually changed the biochemistry, meaning is the protein changed? We can actually do that. This is the kind of Western blot Curtis mentioned earlier. You can actually use this assay to detect the biochemical consequence of a variant. And this is kind of a circadian clock. So every single cell has a clock. It's ticking whether you're not recognizing it or not. So that clock is ticking. In this case, we plot it, longitudinally. We cannot see the clocks working. We actually put, biochemically put a hand on the clock to actually observe the clock running, right? With mutation in the mTOR gene, right, you can have reduced, the clock is not running very well. This is like a healthy 20-year-old. This is like a not very good 80-year-old person, And what's really important here is that a little bit of the rapamycin, this 2.5 nanomolar molar rapamycin, that's the dosage that's crucially important. Too much, too little are not good. So this 2.5, 3 nanomolar can actually improve the clock function. So if the clock runs good, well, then you probably be healthier. So that's what it means. Dr. Kruger talked a lot about the TSC, so I'm gonna segue and really hop on his train here. That is, TSC is upstream. It's like a brick, holds a brick on mTOR. If TSC is mutated, its function is lost, it's gonna release the brick, so mTOR becomes constitutively hyperactive. So this is the case. This is condition. This is measured. See here. This is an mTOR mutation here, and this is a TSC2 knockout, meaning you remove the brick. Okay. So you see hyperactivity at all times. This is tested at basal level. This genetic, mutation in TSC causes hyperactivation of mTOR, whether in basal conditions or in rich conditions, meaning basically you remove the brick. It's on at all times. That changed the clock as well. So it changed the amplitude. The clock is not running very well, and the clock is running very quickly. So how do we sleep, actually? So there are two processes regulating sleep. One is you get up in the morning, 10 hours later, you feel tired. And your clock is running at the same time. So both processes say, okay, it's time to sleep. That's how it works, okay? Here, I'm gonna tell you what so several labs, including Joanna Chu, we actually talked about this yesterday. We came to the similar conclusion, all right, independently. So we're working on this. So here, what it says that there are several cell types in the brain. This is the neurons. This is a cell called the astrocytes. It's one of the glial cells in the brain. These two cell types actually communicate. Look at the literature on one of the groups studying TSC mutant mice showed similar things. So as perturb of the mTOR function in the astrocytes actually causes sleep problems on the bottom, okay, and seizure. This is the seizure events in the mutant mice. So I think we are onto something. That is, if we can study the mTOR function specifically in the astrocytes in the brain, and in addition to fundamental cell biology, what happens when SKS is mutated? What does it do to the cell, and what does it do to the astrocytes, and how does it change sleep function? So we're hoping we come to that point quickly. So this is improves understanding at the fundamental level in cell biology and sleep. So takeaways, right? Number one, we propose that using circadian rhythms and sleep as a biomarker, meaning if you are not sleeping well, then it's probably not good, okay? So if we can improve sleep, we probably are doing the right thing in cells as well. So using this as a biomarker and as a therapeutic target, by improving sleep, we may be improving overall health of a person. So we continue to leverage our cell and mouse models to study what happens when you mutate mTOR. What does it do to the cells? Can we find mechanistic targets to improve cell health and improve sleep? The hypothesis is a aspiring one. We want to actually, after decades of research in sleep, we still don't understand sleep very much. Maybe we have a niche here. By studying mTOR function in astrocytes, we may be able to provide some mechanistic insights as well as targets to improve sleep. And finally, this mechanistic, as a basic researcher, we push, march on. We leave these physicians to do what they can, but we want to march on and to see if there are additional mechanisms and additional targets that we can leverage in future research and medicine. So we want to be able to bridge the divide between mechanism and treatment, hopefully to improve fundamentally cell health, improve sleep, both as a biomarker and as a therapeutic target, and overall improve health in patients. That'll do. Thank you very much.